Hi, my name is Tim, and I'll be talking about the role of collagen in the extracellular matrix. So, an overview of collagen is that it is the major structural protein in the extracellular matrix, and since it's the main structural protein, it is the most abundant protein in animal tissues. Collagen makes up about 25 to 35% of our protein count in the human body, and collagen contributes to the mechanical properties, organization, and shape of our tissues. There are at least 28 different members of collagen that contain at least one triple helical domain of the three polypeptide chains, and these chains are wrapped together or bound together like a rope. So the structure of the proteins, like I said earlier, is that it's wrapped together in a rope. Um, these chains are called alpha chains, and these sequences usually contain glycine, X and Y, with X often being proline and Y often being hydroxylproline. These proteins, proline and hydroxylprotein are used for stabilization due to their ring-like structures, or not ring-like, but ring structures. The glycine in the third spot allows for the tight packing of the chains together, and collagen is synthesized in the cell as procollagen before being secreted out and cleaved by other enzymes, and also modified, usually glycosylated, by extracellular proteins. And here you can see, in the picture, you can see the cleavage sites of the procollagen. Procollagen also collagen has to wait for the binding of heat shock protein 47, and these proteins help stabilize the formation of the triple helix. Collagen is found in the extracellular matrix. It is the basic structural component of connective tissues and contribute to the architecture, shape, and mechanical properties of tissues and it ranges from the tensile strength in your skin to the traction resistance in ligaments. Collagen also has fibril associated proteins that bind fibril collagen proteins to each other or to other matrix components. So these associated proteins are generally used to bind the collagen proteins one end of one protein to the other. Some of the collagen has frequent interrupts in the glycine X and Y repeats to allow for more flexibility to bend as network forming collagen. And so these collagen are less stiff, I guess, compared to bone, and then so these collagens are more likely to bend like our skin. Collagen participates in cell matrix interactions as ligands of integrins, DDR1, DDR2, GPV6 or GP6, and LAIR1. Many diseases are caused by a mutation in the collagen alpha chain code or code sequences, and osteogenesis imperfecta, or OI, is one of them. It's a generalized disorder of connective tissue that results in fragile bones which break easily. And most cases of OI are caused by a mutation in the type 1 collagens, COL1A1 and COL1A2. And about a third of these mutations in the 1A1 sequence are lethal, with 2 of the sequences aligning with major ligand binding regions, meaning that since these sequences are mutated, the ligands are they're mutated and they're unable to bind with whatever receptors. And mutations in 1A2 are mostly non-lethal. So some recent news involving collagen is that this paper explained that the process in which collagen binding partners recognize their binding sites in the fibril, or the protein, is not very well understood. And so this article approaches this by looking at the receptor binding sites of the collagen and the surface of the collagen from X-ray fiber diffraction overlaid with a collagen 1 fibril observed by the scanning electron microscope and AFM. And so they used this information to build an atomic model, and SASA calculations were performed to find the partner binding sites. They came to the conclusion that cleavage of MMP allows for interaction sites to be available and is important for fibril turnover, tissue repair, and development, and that due to the nature of collagen, the SASA calculation only accounts for a single moment in time where some of the binding sites are available or some of them are not, while when instead, since it is constantly undergoing motions, the binding sites that are available changes at any time. So a question I had about collagen is, what are some potential areas of interest where further collagen research would be beneficial? Thanks for watching.